When we talk about oxygen binding to hemoglobin, we use something called an affinity curve. And this affinity curve basically tells us that at what partial pressure of oxygen does the hemoglobin become saturated. So we can graph percent saturation of hemoglobin versus the overall partial pressure of oxygen. Now, this curve is shaped in such a way that it looks a little something like this. Right? And so what you see is that at small changes in partial pressure that you get sort of these large changes in percent saturation. One of the reasons that is because as an individual oxygen molecule binds to hemoglobin, it increases the affinity. So when one binds, it makes it easier for the second to bind, and when two combine or bind to hemoglobin, it makes it easier for the third to bind, so on. Right? And so this is known as the oxygen hemoglobin affinity curve, or some people refer to this as the oxygen hemoglobin saturation curve. Right? Now, there are certain things that can alter this curve. So if we look sort of at like normal circumstances, right, we get a curve that looks something like this. Right? Now, if we alter the pH of the system, right? Let's say that this is here is at a pH of 7.4. Okay? If we make the system a little bit more basic, it's going to shift that oxygen hemoglobin affinity curve to the left, right? So this is at a pH of 7.6. If we make it a little bit more acidic, we're going to shift it over this direction, like so. So this is going to be a pH of 7.2, right? So what does that mean, right? So let's say that this here marks 50% saturation, right? And we'll say that at 50% saturation, we need the certain partial pressure. We'll call that X. But if we want at a pH of 7.2, we want to maintain that partial or that saturation of 50%. Notice that the partial pressure of oxygen needs to go up. So in order to maintain that same level of saturation at a more acidic pH, we have to have a higher partial pressure. The opposite is true if we want to maintain that 50% saturation for a pH of 7.6, right? We need a lower partial pressure than normal. Okay, so that means that at a more basic pH, it's easier for oxygen to bind it to hemoglobin, and at a more acidic pH, it is harder for oxygen to bind to hemoglobin. Another way to think of this is that at this low pH, this acidic environment, it's actually easier for oxygen to be offloaded from the hemoglobin. So think about what's happening inside of your tissues, right, that are undergoing cellular metabolism. Those tissues are becoming more acidic. So where it's more acidic with that lower pH, right, we want to make it easier for our hemoglobin to release that oxygen so we can deliver it to the tissues. So inherently, that's going to make sense. Okay, so pH can shift our oxygen hemoglobin affinity curve. How do you think changing temperature might impact this? So if we use a higher temperature, right, if we increase our temperature, it's going to shift the curve to the right, right? So this is going to be an increase in temp. If we lower the temperature, we're going to shift it to the left. So this will be a decrease in temperature. Again, this is going to make sense because if we say, okay, here's our 50% saturation, we need X partial pressure, right? At a warmer temperature, like at our tissues where we've undergone lots of metabolism. Think your muscles, right? That have increased the temperature of our muscles. We want to have a higher sort of partial pressure because we want to promote that hemoglobin releasing that oxygen. So if we think about these sort of things inherently, they make a little bit of sense, right? So what if we looked at sort of, here's our normal, right? What if we looked at the partial pressure of oxygen here, right? And what if we said that this was at a partial pressure of carbon dioxide of 40 millimeters of mercury? What would happen if I did 80 millimeters of mercury? Right? 80 more CO2, so it's going to shift it this direction. Why is it going to shift it that direction? So this would be 80 millimeters of mercury for partial pressure of CO2. Well, if we have more CO2, we want to make it so that our hemoglobin releases our oxygen, right? Because this is what's going to happen at the tissues. Okay, so if we decrease the partial pressure of carbon dioxide, take it down to say 20 millimeters of mercury, it's going to shift it in the other direction. Okay, there's one more thing that can alter or shift our oxygen hemoglobin affinity curve. And so here is sort of normal. And this is a molecule known as 2,3-BPG, which is 2,3-bisphosphoglycerate. You'll also see it called 2,3-DPG or diphosphoglycerate, but that's sort of the old school term, right? Now we call it the bisphosphoglycerate. And so this is a molecule that is an intermediate in the glycolysis pathway, right? So if we increase our 2,3-BPG, right, we're going to shift in this way. Why do you think that is? Think about how we've looked at all the others, right? Well, if we increase glycolysis, right, glucose breakdown, right, we're going to increase our 2,3-BPG at the tissues where we're doing all this work, and we want to make it so that we have a higher partial pressure of oxygen needed to reach saturation, or we want to make it easier for it to release, right? So this over here, this shift would be a decrease in our 2,3-BPG. Okay, so 2,3-BPG is something that actually accumulates at high altitudes, right? So if you go train at high altitudes, you can get your body to basically have a higher natural level of 2,3-BPG, which makes it easier for you to unload oxygen at your tissues. Okay, so all of these, right, are shifts in this oxygen hemoglobin affinity curve, and shifts in that curve, right, are known as the Bohr effect. It's known as the Bohr effect. A lot of people, though, will limit the Bohr effect, right, to changes in pH that shift the oxygen hemoglobin affinity curve. Okay, so there are sort of, depending on who you talk to, right, the Bohr effect, they might refer to any changes or shifts in the oxygen hemoglobin affinity curve, or they might specifically refer to pH.